past decade, the number of N. numeri jellyfish occurring in the Yellow Sea near China has increased dramatically. Therefore, the number of hospitalized patients for the stings of these animals has also increased. Jellyfish stings are incredibly painful and, if severe enough, can cause long-term scarring of patients. This paper demonstrates the use of tetracycline, a metal proteinase inhibitor, on the dermal reactions caused by a jellyfish sting. Metal proteinase apparently is linked to the venom toxicity and, more importantly, to the dermal reactions. It is therefore easy to assume that a metal proteinase inhibitor would decrease the inflammation caused by a jellyfish sting. In order to test this, the scientists tested the effect of tetracycline in both in vitro and in vivo models. To do this, they collected mature members of the jellyfish from the South Sea of Korea, and then dissected and froze tentacles to collect venom samples. Then they cultured both HACAT, or human keratinocyte, and NIH3T3, or mouse fibroblast cells, to determine the toxicity of these stings. The cytotoxicity was determined by measuring the mitochondrial dehydrogenase activity in both boiled and non-boiled specimens. The results showed that the venom was more degenerative to human cells than mouse cells. However, both had significant loss in viability over the course of the trial. The next test was performed on rabbits purchased from a clinical store. These rabbits were treated with respect to the Ethical Code of Korea. 2 tests were performed on the rabbits. The first was that each rabbit was injected with 500 units of venom, and then one group was treated with straight lanolin cream twice a day for 48 hours. The second group was treated with lanolin cream plus 5% tetracycline cream. The results for this and the following experiment were very impressive and exciting. The second test was a premixed venom test where the jellyfish venom was incubated with tetracycline, and this mixture was then injected into the animals. The size of the skin lesion was then measured at 1.5, 6, 12, 18, 24, 30, 36, 42 and 48 hours respectively. The results showed improvement among most of the group, and the graph demonstrates this by the 2 tetracycline lines, that is, mixed with venom, and external cream, having the least area of lesions other than the PBS group. The graph demonstrates that tetracycline works best if incubated with the venom. However, it is still dramatically more effective when used as a cream compared to the lanolin cream. In summation, the use of tetracycline on jellyfish stings is showing promising results in the laboratory and can now move forward into the next stage of development. Hopefully, this cream will become a viable option for the treatment of severe jellyfish stings worldwide. Thank you.